start and use a couple of minutes for introductions and stuff. So maybe people are still coming in. So I'm Ville Meretoja. I'm a facility manager at UT Health Flow Cytometry Service Center here in Houston. It's my pleasure to have Mike Stadnisky from Phitonex to give a presentation under our FlowTex umbrella. Today, Mike is going to speak about their new dyes, their technology, and how to apply them to make a really multicolor panels, 40 colors, and even more. Before we start the webinar itself, I want to give a little advertisement for forthcoming FlowTex seminar or the Congress or however you want to call it. It's going to be in February 2021, and this year it will be all virtual online. The plan is to have 2 days, 4 hours each, different topics and educational and scientific content. Please go to our FlowTex website to see updates, and also on that same FlowTex website there is lots of links to our previous webinars, and we update that constantly. So you can see our links to archived YouTube videos and recordings of our webinars, and also this webinar will be archived in there in the end. It's my pleasure to introduce Mike Stadnisky. He has a long background in flow cytometry and in BD and FlowJo. If you say a couple of words about yourself and your company, and then start your presentation, please.
Yeah, great. So I'm Mike Stadnisky. I'm the CEO and President and CEO of Phitonex. I used to be CEO of FlowJo, and after that, VP GM of Informatics with BD Life Sciences. And you know, I'm really excited to share this with you today. And we'll lead up to this question, but I'll have you thinking about it now, which is, what if you could design a fluorescent label from scratch? And think about that more as an engineering problem, and then what would that mean? And so we're gonna tackle that in multiple different ways today. And this is FlowTex edition, so I've brought both new slides, new data, as well as my cowboy hat, just in case I need it. So, but no, I'm very excited to show you what we've been up to. So the first thing though that I wanted to put a pin in was, what is conventional dye performance, right? Why is the next 45 to 50 minutes important to think about? And then I'll add what we've been looking at as our North Star when we think about how we design a new fluorescent label. And the fact is that conventional dyes, in fact, only perform in 2 different ways, right? And I'll show you what that means in a minute. And both of them lead to spread, which I think we really need to key in on that, which is a loss of data resolution, the inability to resolve, or the difficulty to resolve, different markers, right? Something I was obviously, very obsessed with when I spent most of my time doing data analysis, and realized really at the end of the day that it came down to our underlying fluorescence technology. By the way, just as a housekeeping item, if you have any questions, please do drop them in live down below into the Q&A, and Bill will interrupt me, and we'll make it a little bit more interactive. But those 2 different types of conventional dye performance are one that we're all very familiar with, because we are subject to this quite a bit, is the PE and PE tandems, which are cross-excited by multiple lasers, right? In fact, we even see this manifested in our hardware, where we have only a few number of detectors, for instance, off of an LSR Fortessa, off of the blue, or where we go on to run a panel on a Cytoflex or a Symphony or any instrument, and we're not able to leverage all of the detectors off of the blue. And then the other type of characteristic we get is something like the brilliant dyes, where we might get narrow excitation, but we have incredibly broad emission, right? And you might think, well, okay, that's fine. I can handle these kind of things that lead to the crossover at the detector level by using compensation, right? So let's just take 2 traditional dyes as an example: APC size 7 and PE size 7, right? And if you look at this, right, you can say, okay, well, clearly they've got some emission overlap. You can see there is quite a bit of the APC signal, right, which is crossing over into the PE size 7 detector. But you think, well, that's fine. I can compensate that, and I can deal with it. The problem with that is that after compensation, you see this manifesting as spillover spreading error, and you do see this as a loss of resolution in the data. And let's also be clear about what this is. These are false positives, right? And so it's this spillover spreading that really leads to a huge impact on the loss of data resolution, something that we've used as our North Star. And I'll describe how we look at this both subjectively and objectively in the context of new floors. And what we're really trying to head towards is the ideal scenario where you don't have this loss of resolution and you could separate every marker that you have and every fluorophore that you use in every channel. And, you know, I was searching the internet for really good examples of, okay, well, how do you explain this kind of measurement error that leads to spillover spreading that you actually see that propagates itself through the map that you see in your data here? And the answer came to me when I was thinking around FlowTex and the number of bulls that you guys have in Texas. And the answer is, look, even though this is from Pamplona, look, if I were to start the running of the bulls and I told you people were running incredibly quickly and I told you to start counting every single person that was running by you, at some point you would just lose count, right? And you would not be able to count everything else. Now, instead of them being people and instead of this being you, if they were photons and I called you a PMT, you'd have exactly the same problem. So as photons are hitting you and you're trying to count them, you're going to end up with some measurement error. And that's exactly what's happening. And that's what you see when you get this loss of resolution in your data. And also, you know, to break the ice a little bit, why does resolution matter? Well, resolution loss in our data really matters quite a bit. So here's 3 good examples I think that show this, right? So if you have a resolution loss of 20%, you're not able to determine the difference between a bagel and a sleeping dog. If you have a resolution loss of 60%, you're not able to determine the difference between a blueberry muffin and a chihuahua. And if you have a resolution loss of 90%, you're not able to determine the difference between a pile of raw chicken and our former president's face. So with that ice broken, hopefully, I'll ask this question again, which is, what if you could design a fluorescent label? And what if you could think about this notion of being able to resolve markers and doing that across the entire spectrum, across all the fluorophores that we use? And given that, what would panel design mean? What would be the way that you would view how we design panels? I think the 3 requirements that we would come out with and the 3 things that I'll attempt to satisfy using data are that labels would be bright and clean. They wouldn't have the spread characteristic, and they would narrowly excite and be narrowly emitted. They'd also be silent and violet. We could leverage all that space for what we want to do with violet labels. Obviously, we limit cross-laser excitation. In fact, we make that near 0. The second part of this is it's not just about new colors or even their performance. They have to be incredibly consistent and incredibly stable. Notably, they should be way more stable than the tandem dyes. We'll talk about that as well. And the third part of this is that is sort of like a dream feature, right? Which is, well, what if we could control the brightness of our labels and actually tune that to the underlying antigen density? So I'll actually show you we have the ability to do that. How do we do any of these items? The answer is we take small organic dyes, bind them onto oligonucleotides, and then can actually assemble them into this DNA structure called the phyton. And it's this scaffold that enables us the exquisite engineering of new fluorophores, both in terms of the components that we choose to put onto the scaffold, but also the fact that we can change the distance in between the way that these dyes interact with one another. So we can leverage fluorescence resonance energy transfer on the platform. And since that scales with the 6th power of distance, we can actually tune, again, the excitation and emission to actually get FRET on the platform and have that be incredibly highly tuned, right? And so I can actually use that to tune excitation and emission. I can also tune brightness because I have 4 independent arms of this structure. We've also done some work on tunable signatures that I want to show you today. It also enables me a lot of flexibility because I can hang an oligonucleotide off of the end of this label and attach it to antibodies or other proteins. And one final thing I'll mention here is that while we're leveraging FRET, these are not tandem dyes. And in fact, as we'll discuss, you can leverage, when you think about compensation controls for low frequency antigens or low express antigens or low frequency populations, you can actually compensate these using CD4 or CD8, whatever, because the fluorescence doesn't change. They're held rigidly on this very stable DNA platform. What have we done with that? And so I'll show you what our approach is. But the headline is we've released 17 unique fluorescent labels, 17 unique colors across 19 labels in 2020. And so I'll show you what that looks like. But the approach that we've taken is actually to, again, think around excitation and emission and think about this as a pixel by pixel exercise where we can narrowly excite, which I'll show you an example of in a moment, and narrowly emit, and then get high resolution of the markers on which these are on the back end. So we really think about a flow cytometer as something where we can actually go in pixel by pixel. The other thing that we also like looking at, and so we'll typify this in a moment, is what a traditional dye actually looks like across the entire spectrum. And it's funny because actually it looks like someone's actually annotating over my screen here. But in spectral cytometry, a dye signature is actually the result of non-ideal dye performance. So we actually like looking at these spectra, right? We've got this primary detector here, right, which is where this fluorophore is intended to emit. And everywhere else, if you think about all this secondary channel fluorescence is adding spillover spread. It's leading to a loss of resolution of the data, right? So while it increases the theoretical limit of colors in the panel, right, we cannot outrun spillover spreading. We can't outrun the loss of resolution of the data, right? And in fact, to that end, what would a perfect dye look like? And a perfect dye would look exactly like this. It would only emit in about a 10 nanometer range. It would hit only the detector that's desired. It would contribute no fluorescence anywhere else or spread, right? And so you wouldn't need to compensate it. You wouldn't need to worry about spillover spreading. And moreover, when I show this, people say, I would also like to be able to tune the brightness of that, so that if I have something that's very, very low antigen density, I can move the brightness up. But if I have something that's very high antigen density, like CD45, we should tune that down. And if you titrated it down, you had a tad less than perfect blue, you would actually have this kind of signature. And so let me show you how that works with some of our examples of some of the labels that we've made. So the first set of labels that we came out with were off of the blue and yellow green laser. And the intent here was to be able to stack the yellow and green lasers as well as the blue lasers and actually move away from things like PE dazzle and be able to leverage every detector that's off of those lasers. And in fact, the way we can do that is if you look at PE dazzle, it's promiscuously excited, right? Off the blue, the green, and the yellow lines. And we said, quite simply, well, what would happen if we emitted if we had more fluors that were simply only excited off of the blue or the yellow lines? And that's exactly what we've done off of our first set of fluors, where they're both narrowly excited and then can also emit narrowly, right? And so we developed these actually, interestingly enough, on an attune. And the whole goal was we want to be able to leverage every single detector off of that attune instrument. And then I'll show you that we were able to step that up and really be able to push the limits on flow cytometry and think about how we characterize these, right? And so we've come out with this 6 set of labels of the 17 unique colors. And the way we name these is based on their excitation, blue, and then their emission, 530. So this would be something like a replacement for FITSI. And so I'll describe kind of how we like characterizing these, and I'll show you some examples of that. But the way we like characterizing these is always through the lens of where we're trying to go in panel design. And the goal is, of course, to separate as many populations, as many markers as possible. And what that leans on, again, is this resolution of the data. And so what do you need? What are the ingredients to make that happen? Well-maintained and QC instrument. Can't help on that front, right? The second part, though, is understanding spread and how to deal with it, right? And so like literally thinking around the data resolution from the very beginning. And so we'll describe how we engineer for that through 2 different levels and an appropriate set of controls, which we make a lot easier because unlike tandem dyes that have the instability that we'll describe, we don't have to worry about that, right? And so the question we have is, well, what if we approach this from 2 different angles, right? What if we attack both the fluorescence and made that incredibly clean, right? And I'll show you the characterization of that, but also at the panel level. And so I'll show you how we think around panel design as well, using the North Star of spillover spreading, right? And spillover spreading, in fact, is the enemy, without a doubt. This is what causes the loss of resolution in our data. And in fact, if we look at 5 logs in a range of detection that we can do in biology, it's really spread, or sorry, on our instruments, it's really spread that is the largest contributor of background here, right? And as we've said, this is what manifests itself in our data, right? And this error manifests itself in a loss of resolution. And in fact, one of the simplest plots I'll show you, but one of the possibly the most sort of impactful, is the fact that if you look at our labels, and this is looking at it on our attune instrument, and this is off of adjacent detectors, what you're seeing is honestly this ideal dye performance where not only are the medians lined up post-compensation, which is what you'd expect, but also the standard deviations are lined up as well. So in fact, you are getting very, very high resolution of these markers, even though you're in adjacent channels. And so the way that we like to characterize this resolution or loss of resolution, as it may be for traditional fluors, is we actually take a look at all the dyes that we have in an experiment, right? On a y-axis, and all the dyes we could have on an x-axis. And we look at the sum of these. And we identify fluors that cause a lot of spillover or a loss of resolution. And that's the way we objectively look at this. So again, going back to some examples here, right? This is actually taking a look at our these first 6 fluors, which we're using as an example, post-compensation. And again, you would expect to see, you know, if you had a lot of spill, you would expect to see this spread in the data. In fact, you don't see that after compensation with these fluors. So subjectively, we can see, okay, well, there's something going on with the performance of these fluors. Right, there's something to this fact of limiting cross-laser excitation and having very narrow emission. I'll show you some examples now of how we benchmark these labels. This is taking a look at effect symphony of us stacking up Nova Blue 610 versus Brilliant Blue 630. And you can see that while we're not as bright as Brilliant Blue 630, although put a pin in that because I can actually control the brightness of this label, and I'll show you that, we're notably very quiet in the violet, in the UV, and the green. So as you'd expect, right, again, and as I'll show you, the spread performance of this dye is really, really good. Similarly, we've got very bright labels off the yellow. This is an example of one of our dyes, Nova Yellow 610, again on a Symphony, comparing it on an apples to apples basis, right? We're looking at it versus other labels that you could use in the same primary detector like Elixir 694, PECF 594, ACD, and it's intended to replace the use of these labels. And the reason to do it would be, well, it's in the data, right? It's brighter than Elixir 694, nearly as bright as PECF 594, brighter than ECD. But again, it's not all about brightness. It's really about cleanliness, right? Because we're incredibly clean in the UV and the violet as well as the blue, right? And the way that cleanliness, that spectral clarity, or other people have referred to this as spectral hygiene, can manifest itself is actually when we go and we look at, again, looking at the impact of resolution, where the lowest score would be the lowest impact on the resolution of the markers, right? We were trying to achieve the lowest score there, right? And in fact, that's exactly what we see, right? So when we sent these labels off to the National Institutes of Allergy and Infectious Disease, they looked at our Nova Yellow dye versus 30 other traditional labels. And while, as you saw, it was bright, it had the lowest spill, right, versus 30 other dyes. And this blue dye had a very good performance versus the brilliant blue dye. Again, thinking about it in terms of we want to achieve very, very low spread. And it's not just us that's seeing this. The way, the reason, for this low spread and the thought around replacing these is to enable people to do exactly what Oliver Burton's doing at the Babraham Institute, right? Which is that these NovaFluors show more restricted emission and excitation profiles. And in his case, he's going in and replacing these whole hog to think about how he can leverage clean fluorophores in his single cell CRISPR based assays, right? And so he's seeing that here, looking at this across the spectrum, Nova Yellow 570, clearly a much cleaner replacement to PE, and really enables quite a bit more in terms of the ability to phenotype and leverage, honestly, both the blue and then other lasers besides, because I think one of the things we also sort of discount sometimes is just how much PE is emitting into lasers that are very, very far afield. The other thing too is, they're much, much cleaner than any of these dyes that have come out, particularly in the blue and the blue yellow and the yellow green from BD, which is again no surprise because these are engineered from the ground up to have this kind of performance. And so the question that comes up, of course, is like, okay, well, how do you implement these in panels? Do they work with the brilliant stain buffer? And if you could push this to its absolute limit, what would that mean? And how could you think around characterizing your labels? And what would we make of that? And so what we did is we took this 35 color backbone panel that was shared by Cytec. We got the 34 of commercially available antibodies, and we quite literally dropped in 6 of our colors, right? The six that I'm using as an example here. And we looked at them on both high antigen density markers like CD19, as well as more variable and low antigen density markers like some of these chemokine receptors. And so one of the big things that we had, again, thinking about this North Star of data resolution, is we wanted to ask a question. At the upper limit, where we're pushing this as hard as we can, right? This is up at 40 colors, right? What's the impact on resolution when we can take one of these colors at a time and actually characterize that? And so what we did is we looked at the spillover spreading matrix of the 34 color backbone, right? Because that's the hand that we're dealt, right? This is what we're dealing with. We're not changing up the backbone here. But Sedna and our team ran a really elegant experiment where she dropped in one color at a time onto that backbone and then looked at the impact, right? And of course, an ideal fluorophore would have 0 spread impact. It would have 0 resolution impact on any of your other labels. And what's interesting about that is that if you could achieve that, you could be in a situation where you were designing your panel, and as it always happens, right? You design your panel, you're ready, you've optimized it, and then you keep reading papers or you go to Journal Club or you see something new and you want to put 1 or 2 or 4 more markers into your panel. What do you do? And the answer right now is, well, you can't do much. But if you had something where you could add colors and have essentially 0 impact on the resolution of the markers, you could upgrade your panels essentially on the fly, right? And so again, what we're looking at here is essentially impact on resolution. So what we're trying to aim for is a graph that's flatline, where there's 0 impact on the resolution. So what we see here is when we put this we put Nova Blue in on its own, we see that we get an increase in spread for BB515 and 15. We're looking at this change in spread, which is an incredibly challenging combo, right? And we're really cramming these 2 detectors in the blue, and there really isn't possible to do much more there. So as I'll show you, we'll actually drop BB515 out of a really high color panel, 45 colors, and we are able to resolve and separate those populations. But what's way more important than that is what you're not seeing in this graph or the following two graphs. And in essence, you get spillover-free upgrades in this panel. So you're able to put both Nova Blue 610 and 660 into a panel and not add really any spread at all, no loss of resolution to your other markers that are on these dyes. And that's been really, really powerful, right? So we've got folks now who have been able to, for instance, test this on their antibody using our conjugation kits, and then they'll actually work this through into an antibody printing service that I'll show you towards the end of the talk, because now you can quite literally upgrade your panels and leverage these clean floors. The second part of this was like, okay, well, how does that work on some of these yellow dyes? Because you also drop those in as well. What we see is some increase in spread, right? Some loss of resolution when for Nova Yellow 570, for instance, with the dye it's intended to replace, PE. So it's interesting because what this shows you is that in a spectral system, you could use them all together, right? And get very, very low impact upgrades to your panel. And in a conventional cytometer, what you're seeing is, in fact, these three yellow dyes have much better performance than PE, PE Dazzler, PE Sci-Fi, which you'd expect, right? Because these are high spread dyes. And we'll show you in a moment, where she characterized that and actually took PE Sci-5 out, as well as BB515 out of a panel we were working on, right? So low spread, low impact, higher resolution of that biology. So I want to show you how we push that a little bit further, again, thinking up at the upper limit of what's possible here as a way of characterizing dye performance. So we shared all of the raw data for this experiment, the deconvoluted data, as well as the analysis. We also came up with this subway plot, which took on a life of its own as a data visualization tool. It's actually sitting here behind me. But the point of this really, well, one, other than communicating the data out and the phenotyping, was also, look, now you have the ability to leverage these clean floors to look at chemokine receptor expression across 32 different phenotypes and do it with, very low spread impact to the rest of your panel. And this, to me, it's incredibly exciting because as we look across this experiment, we look at a 45 color experiment that I'll show you in a moment, we're able to do both broad and deep immune phenotyping. And so what does that mean? And what are we doing with customers now that we can leverage that? So in the immunophenotyping world, right, we knew that, you could do about 20 parameters, right, with traditional dyes. We've already implemented panels with folks where they're only looking at one subphenotype with those clean fluorophores with up to 24, and now they're moving up to 28. And when you have low spread floors, you can examine multiple co-express markers, because if you have that clean sort of pixel fluorescence, you can just add a lot of those all in the same phenotyping tree. The second part of this is actually what, with broad, there's a publication that shows you can do 40 with traditional labels. I've shown you how we do it with ours. And in fact, what we did when we created our 45 color panels, we went back into the traditional dyes and removed, if you will, the problem children that are in there from a dye perspective, and then upgraded it to 45 colors. So I'll show you that now. And we're working with customers now to do 52 colors. The point of this actually is not necessarily to just push the limits of flow, although that's definitely fun. It's also to be able to characterize these dyes and show you that they can be used across any set of instruments, because the performance of the dye is the performance of the dye. And low spread means you can do more across every instrument, right? So we're actually running a number of experiments now across an Attune and an LSR Fortessa, as well as a Cytoflex, where you can leverage every single detector off of those instruments, right? So you're not fighting with one hand tied behind your back in terms of what you can do on those instruments either. But the way that we think about this, that panel, or the way we think about any of these high parameter panels, is exactly the same, which is we set a hit list. And how do we do that? The hit list that we look at is we actually say, okay, what are the worst spreading dyes that are in there? What are the dyes that are having the worst impact on our ability to actually resolve markers? And what we found in this one was that BB510, BB515, and CF568 were the worst performers from a spread characteristic. And so what we did is we took those out of the panel, and we had always had this notion of like, well, if you can remove those, shouldn't you also decrease the amount of spread that's in the panel, right? And the answer is yes. You can actually quite literally decrease the amount of spread that's in the panel. And then what you go is you go back in and you replace those with these high performance floors. As it turns out, one of the surprises that we had was, well, we knew that we were going to go after these three for replacement. When we went back in and looked at, okay, well, how are we looking across all the spread now? Let's go back in and regenerate our hit list. Again, what's the highest spreading dye in here? We see the PE Sci-5 is the next target, right? And we know this because it's emitting into the blue, it's emitting into the yellow. And so we also went in and removed PE Sci-5. And so that's what we did. So we went in and dropped all four of those dyes out, and then came back in against those same antigens with our clean floors, right? And then because we've got this clean fluorescence, we also then upgraded the panel to be able to leverage more of the detectors, again, on this instrument, right? and this is a 64 detector instrument, right? But the analog is the same for any instrument. The fun part about this experiment, of course, people are always like, oh my gosh, that must be a pain to analyze. And I've got to be honest with you, it really wasn't, right? Both due to the fact that the experiments were really consistent, but also the fact that the instrument was also quite consistent. And so we were able to actually take the phenotyping gates, right, all 32 very broad phenotype gates from the 40 color and quite literally drag and drop them onto the 45 color data and use that as the base to really phenotype this data out. And I think the fun part of this, beyond just the analysis of the data, is that we're able to use fluorophores that are anywhere from 20 or 10 nanometers different than one another. We can do a couple of different things with them. One is we can phenotype very broadly. We can look at a broad set of these phenotypes. So we can, for instance, we can look at, okay, this is where the B cells are, right? We've got here fluorophores that are only, in this case, 20 nanometers different, that are both excited off the yellow. And we're able to uncover new biology with them. So when you have that notion of clean fluorescence, you can actually leverage the full capabilities of your instrument. And of course, you can do that for very broad phenotyping, or in source of another talk that I gave, where we actually can look at this and then slice and go at different layers with the T cells. Because again, when you have 45 markers, you can go both broad and deep, leveraging spread to really drive that. So the second part of this is like, you present colors, present performance, and people generally like, that's great, but how do they work in the real world, right? And this became even more of a, this went from a comment to like a shout, right? And I think it's because we've been dealing with some of the same issues as we purchased a huge number of antibodies as we deal with the instability of tandem dyes. So I'm gonna shed some light on that, and then shed some light on also how we think around changing the game, if you will, around both consistency, what do we get when we purchase antibodies, as well as stability. In other words, like how do we deal with the things that we've inherited from tandem dyes? And so, tandem dyes are I think, and we all know this, are fraught with issues, right? And so they exhibit instability in solution, right? And so we'll see that again manifested in a moment. They decouple differently on cell phenotypes. Really interesting paper from 2009 that actually shows that you can get 40% degradation of APC size 7 and APC H7 after about 30 minutes on a monocyte, and that's actually quite a bit different than a lymphocyte. One thing we've also focused very heavily in on is that these labels break down very quickly in fixative solution, right? And so this is actually some data from Paul Wallace here, and what you're seeing is the degradation of the CD45 APC size 7 into APC, right, where the size 7 is essentially being shed off after 72 hours, just in storage and fixative in a fridge. That's it, right? And 24 hours after that, of course, you essentially have no more tandem dye. The second part that we really wanted to attack as well was that tandems, in our observation, show that polymer dyes exhibit significant lot-to-lot variability, right? And another, of course, fold in on this because of this instability, right? And because of the notion that every time you attach a tandem dye to an antibody, that fluorescence changes, is that it's a pain. Let's just be honest, it's just a pain to compensate these things, right? But if you really dig in on this, you start scratching the surface here, some bigger worries that come about, right? Okay, we can all accept that you can't use two different tandems and that are on antibodies from different vendors. Okay, fine. You can't use two different specificities. Okay, we get it. We know this, right? This is what we're taught in our flow classes. You can't use two different lots. Okay, now we're starting to get a little bit like, well, that's kind of worrisome, right? You can't use one that's fixed versus unfixed. Okay, not as worrisome. Okay, you can't use a lot that's newly purchased versus one that's 1 month old. I'm sorry, what? That to me is absurd, right? And so it shouldn't be on us to determine, to look at this and why is there that much variability in these tandem dyes. So to the point, what we've done is attempt to push the limits on this, right? And really see, okay, well, if you have this incredibly stable, this rigid DNA backbone that you can attach to antibodies, just how stable are the fluorophores? So I'll tell you that in some data that I'm not showing here, that you can take our labels, the fluorescent labels on their own, leave them out on a bench, and they're spectrally stable for at least 2 weeks. In this case, we're gonna push that harder and say, well, what about the conjugated antibodies in fixative? So what we did is stain T cells with a CD3 that was conjugated to one of our labels and left them in 2% paraformaldehyde for 2 weeks. We didn't fix them and then come back with PBS. We just literally left them in fixative for 2 weeks, right? Which would absolutely destroy a tandem dye. So on the left here, I'm just showing you some fresh staining of our T cells, right? So this is just CD3 positive cells. And then we went and compared that to those that had been stained 2 weeks prior and left in fixative. In fact, we saw there's no difference, right? And so the question we kept getting is, wow, that's really interesting. It's also spectrally stable. So we just looked at every single detector we had on our attune, which gives us an approximation of across the spectrum how stable these are. And what we're following up to characterize this on something like an Aurora or an 97,000, something that has access and just has more detectors. What we see when we look at this on the attune is it's incredibly stable across the spectrum. So you can leave essentially your stained cells in fixative for at least 2 weeks, and now we're actually testing longer, which I think is also impactful when we think around some of the limited access that we have to our instruments now, where you want to be able to get in there and stain and then leave them. The second part of this was like, okay, that being said, what about room temperature stability as well? What about just the baseline stability of these conjugated antibodies? And so we started down this journey by quite simply leaving our conjugated antibodies inside of a lab bench, light protected in PBS, no special buffers to prevent aggregation because we just don't ever see aggregation. We just wanted to just see how stable these are, right? It's been alternatively called the Europe shipping experiment, the Australia shipping experiment, or the lazy graduate student experiment. And what we see is that there's actually no difference in the conjugated antibodies that were stored in a bench at room temperature, or those that have been stored in the fridge, right? And not only is there no difference in terms of what the staining profiles look like, we also pushed this a step further and said, is there any increase in background at all, right? And so what we did to look at that, and it's one of the metrics that we are fond of, is you could have for instance, you could have the brightest label in the world, but if you're not able to separate it from background, it's somewhat meaningless. So we always focus in on separation index, right? To account for the fact that if you have an increase in background, that you should really be dinged for that. So what we did is looked at the separation index again over those 2 weeks across the time course and saw no difference in the separation indices of these labels that have been stored at room temperature. So again, we want to push that a step further, right? So as we've been purchasing a lot of these antibodies, we see these lot-to-lot variability, we've seen quite a bit of lot-to-lot variability with respect to some of these newer dyes that have come out. And we said, look, that's insane. What we're going to do is turn that on its head, and we're going to share our manufacturing data. So this is actually 2 different lots of our manufacturing data of our fluorescent labels. What you're seeing is what you're not seeing, right? We do not have variability in the manufacturing of these fluorophores. And in fact, our spec is to be within 5% anywhere in the entire spectrum, which is within instrument variability. If it's more than 5% different than essentially our standard lots, we throw it out, right? And so we're very committed to making lot-to-lot inconsistency with respect to fluorophores, a thing of the past, and similarly doing that on the conjugated antibodies. So we also took these, conjugated them to CD4, and sent them to an outside lab and said, go ahead, but please run these across titration. Tell us about, again, looking at the separation indices, because we got to make sure that not only is the signal consistent, but also the background is. And the answer they came back with is that there's no statistically significant difference in the separation indices for these conjugated antibodies across the titration for Nova Yellow or for Nova Blue, right? So the goal, again, really is to make this type of lot-to-lot inconsistency and putting this in on us as researchers, thing of the past. So to that end, we actually published a blog post, which is things to ask your vendor. Can I see the lot-to-lot data on your dyes? Sounds like a reasonable question to me. Anyway, the second part of this too, though, is like, now that you have that kind of stability, right? One of the questions that always came up is like, how big is this thing? Can you get it inside of a cell? Great. So it's actually, this phyton structure is about the same size as an IgG molecule. So it's 150 kilodaltons, right? And it's about 20 nanometers if you lay it flat, but due to the natural curvature of DNA, it has a bit more like a cup structure. And so it's probably about more like 18 nanometers across. And so what we did, though, to answer the second part of that question was we ran some preliminary intracellular and then also some intranuclear assays. And so in this case, what we're doing is gating on lymphocytes. We've got low background here, and this is just a positive control showing you that we can stain interferon gamma PE dazzle. And in this case, we're just titrating up an increasing amount of that antibody. Similarly, low background here, just got the FMO. And in this case, what we're doing is titrating up anti-interferon gamma Nova Yellow 660, and we haven't reached saturation yet, but we are able to get the conjugated antibodies into the cell. And I think it's really exciting. So we're following this experiment up now, because when we have these type of clean fluorophores, we can think around multiplexing inside of the cell and leveraging this to examine a lot more cytokines at one time. Arguably even more interesting, and it was certainly a big challenge, right? One that's come up several times, even with podcasts that are talking about these labels, is, well, hey, this thing's pretty big. Can you get inside the nucleus? Okay, fair challenge, right? And so again, gating on lymphocytes here, looking at the FMO, so low background staining, titrating up this FOXP3 antibody. what you see here in the positive control is that you're able to get, reasonable staining of FOXP3. And in fact, you see actually the maximum fluorescence here is about a tick over 10 to the 4. So interestingly enough, when you titrate up actually the FOXP3 that's conjugated with our NovaFluors 610, you actually begin to start separating the FOXP3 low and FOXP3 high populations. And the fluorescence of these high populations is substantially greater. And so I think a lot of that's due to the fact that, PE is a massive molecule. It's about 240 kilodaltons, right? Versus our molecule much smaller. And we're thinking that there might be greater access to those species in the nucleus. Not sure, but we're repeating this experiment across a larger number of samples and also looking at a number of other transcription factors. But exciting because now you can actually leverage these inside of the cell as well. And the final part of this is sort of like one of those things it's like, boy, I wish I had when I was designing more panels, which is controllable brightness. And so what if we could choose our brightness rather, we all learn flow. It's like PE, like here you go. This is your brightness chart, deal with it, right? And you probably maybe even thought once like, man, it'd be great if I could just choose a color and then choose a brightness relative to the underlying biology that I really need to ask the question about, right? I'd rather be held back by my biology rather than the experiment that I can run. Okay, you're wrong. So we've come up with the first digitally fluorescent labels, and we had to come up with a naming convention for these at the start, which was, okay, well, how do we even name things that have exactly the same excitation and emission, but have different brightness levels? And so what we came up with was the wattage of a light bulb, right? So if you go into a dark room, obviously you need a higher wattage bulb. Same thing for low antigen density, you need something that's higher brightness. So we name these actually based on their separation index of CD4 as we measure on an aurora. And so to give you a sense of this, I think the data is where it's like, for me, that's where the light bulb goes on. You can actually see that there's a substantial increase in brightness here, and it's stoichiometric, right? And you can count on it because of the way that we manufacture these, as I'll describe in a moment. And you can see that, but we now have actually two different versions of NovaBlue 610 and two different versions of NovaBlue 660. And so this can be leveraged in two different ways. If you've got a very low antigen density target, you can choose something that's brighter, or if you have something that's very high in antigen density, like CD45, or in the experiment that I was running, CD44, you could actually tune that down and lessen the impact of the spread of that marker into other channels. So I think we're considering, okay, how do you roll that out when you have a huge set of fluorophores that you're designing across the entire spectrum? And what could you leverage there? We're also looking at more dynamic range here. So we've got some experiments going on, for instance, with exosomes right now, where we have a very, very bright version of a yellow dot, right? Again, stoichiometrically controlled, so it's 4 times as bright, right, as what we offer, sort of baseline on an antibody. What could you do with something like that? So we're also leveraging that notion of what could you do in terms of asking interesting biological questions if you could tune this. These two labels that have this digital brightness were part of a launch where we actually launched 17 unique colors this year. And the way that breaks down, and the way that we're working with people with a very hopefully cool update, is these are the first 6 labels that I showed you. In the context of how we're benchmarking these. We've got those all the way now onto conjugated antibodies and actually have the ability to print those labels, and I do mean print, onto the antibodies that we offer or any antibody that you send us. So I'll show you what that means in a second, because it's kind of interesting. Print, right? And so what we've done on top of that is build out a suite off of the blue to be able to, again, to go in there and think around stacking more off the blue. Same thing off of the yellow laser, where we've also come out with some very, very bright dyes with NovaFluors 690 and 700 to really think about replacing the brightness that you get from a PE to go after those low antigen density targets. And then we've also come out with a set of 4 labels off of the red from APC to APC Sci 5.5, right? And again, the whole point is you can use these together, right? Because they're designed to leverage the whole spectrum, right? And so what do we mean by printing, and how are we doing any of that? So what we actually have the ability to do now, and this is how we make our conjugated antibodies. This is also how we work and hopefully what you'll agree is a pretty new customs program, is we actually take in any antibody and can polynucleotide modify that antibody. And we can set up a matrix by which we can actually print out any of our colors on any of those polynucleotide antibodies. So the impact of that is, okay, well, that's really cool, but what else is a benefit there? And the answer is we're fundamentally changing the way that conjugation works for a tube that you can order from a manufacturer. Right now, when you order something from a manufacturer, there's excess stoichiometry of the dyes, and that's to push that conjugation reaction, right? So it's 5 to 1 or 10 to 1, or because the efficiency isn't very good. Certainly affects cost, but it's also that all that unreacted fluorophore must be removed. Some customs programs just isn't. And this results in a spread of the degree of labeling. It actually results in a lot amount of variation of the number of fluorophores you're attaching to that antibody. And in fact, you can see this in multiple manufacturers' catalogs. It'll say typical FTP ratio is 3 to 7 to 1, right? And it turns out there's actually ends up being this Poisson distribution. There ends up being this large amount of variability in the number of fluors that are on those antibodies. And I kind of want to underline this. That means that there's variability in the amount of fluorescence you're getting on your antibodies. And that's before you've stained a single cell, right? And so fundamentally, right, what that means is you can't quantitate using most antibodies. PEs is the one exception. And it's just that it's so huge that it occludes other PEs from binding. But everyone else is really subject to this limitation. What we've done is come in and develop a system by which we can provide one Phyton label, one NovaFluor on one antibody, right? And we actually certify that as part of our customs program. So again, getting back into this notion of printing and do it in a one-to-one fashion is exactly what we do for us. For instance, we've got this set of specificities that we've got that are in stock, and we can print those across any of the colors that we have. It's actually, for what it's worth, how we make our CD4 kits, which are useful tools in looking at the spillover and characterizing these labels on a set of instruments. And that's exactly what we do. We take polynucleotide modified CD4 and then can print this across a set of colors, right? And then you can run those and use them in flow. We pushed this to its limit, right, and really pressure tested it using the 45 color experiment that I showed you earlier, right? Because we became our own worst or best customer, depending on how you look at it, because we had to validate that we could conjugate all these antibodies, right, to an oligonucleotide, determine the concentration, then print them out on a new color. To give you a sense of how we do that, and it's also how we work with customers in our custom program, is we developed this custom printing process, which we've now used for a number of folks now. And we can take in what we do at first, we take in an antibody, polynucleotide, modify it, we purify it, and then we actually leverage this one-to-one labeling to come in with our NovaYellow 610 dye to be able to get the concentration of that antibody. We run it in flow versus a commercially available antibody using the same club to say, look, it's valid, right? We're staining exactly that same antigen, and we're validating the oligonucleotide modification of the antibody. And then we have the ability to attach it to any desired fluor, right? And then we provide that conjugated antibody back at known concentration and also one-to-one label, right? And so, for instance, for the large experiment, for the large order that we ran, right, we could go in there and again, stack these up and produce a validation matrix that we could not only, print these, determine the concentration, and then be able to print those on the desired color. And that's exactly what we did. For externalizing this, we've got a CD4 kit that enables characterization of any of our labels. We also offer conjugation kits, which enable you to conjugate up to 100 microgram of your antibody. So a number of folks are using that now. Very simple it's a 3-step process to be able to conjugate that, and you've got conjugated antibodies in less than 24 hours, right? And then what we've been working with is that's a great way of testing it. And then when you move up, you can either order some of the specificities that we have or leverage this customs program where we actually print colors onto an antibody, right? So you print us a 0.5 meg of your antibody. We have QC guarantees with respect to spectral QC. We confirm the conjugate by gel. We guarantee no free antibody. We also guarantee that the stoichiometry is dialed in to the point where you're not going to have free dye in solution. And the timeline is a maximum of 3 weeks. So when we do in-stock antibodies, we've been able to go under a week for printing any in-stock antibody on any color, but our customs printing program is for a maximum of 3. So you might be wondering, okay, well, that's a lot of fluors still, Mike. Like, where do I begin? We've published this hopefully interesting tool up on our website where you can actually compare any fluorophores and look at it in the way we've been talking about today. How does it look like on a separation index, and what is the spread characteristic of these labels versus any other labels? And you can look at that also on a per laser line basis, and hopefully a fun interactive way too of looking at the performance of fluors that we're already using and ones that you could replace in there or even upgrade the panels that you've got now. So as I mentioned, we offer CD4 test kits, which are just a great way of putting the hands on the labels, characterizing these on any instrument. We offer these conjugation kits for 100 micrograms for any of our colors. And as I mentioned, we've got this printing program. We've got a number of different labels, but I think the thing I always like to throw out there is, as you can see, we've got 3 of the major laser lines, and we certainly have to go further red, move out towards things like APC size 7. We're very actively working on purple rain. So we're very actively working on labels for the UV and the violet. And so any feedback you have on what you're using for BUVs or BVs or super brights or whatever, all of it is incredibly valuable. So I get even when I get random emails or texts about these labels, all of it ends up going to our R&D team. And it's incredibly exciting to get that kind of feedback because it enables us to prioritize where should we be going and what should we be attacking first off of those labels. So hopefully a fun story about what we can do to essentially change up, if you will, the pace of fluorophore development, as well as the performance of those underlying fluors, and what that might mean for the resolution of our markers and underlying biological populations. My email address is here, myStadnisky@Phitonex.com. Our catalog and our website are up here as well. Catherine, who joins me from Australia, her email address is here. And also you can email info@Phitonex.com, which is an easy way of getting a hold of us as well. And luckily we've got enough time for questions. So please fire away.
All right, are there any questions for Mike here?
Yeah, I've got one that actually came through in the chat. Use that maybe to kick it off. So to two different ones. One is, are you seeing any non-specific binding to myeloid cells? Was the first question. Yeah, it's interesting. So in that regard, we do see binding of some of our fluors to monocyte populations. So if you stain them, you will see binding to a monocyte population, and it mimics what you see with the PE tandems. And so to that end, we've developed a blocking solution that limits that background staining. And so that blocking solution, it's 5 microliters 5 minutes before staining, and then you come back with your stain, and it prevents that background staining that we see, particularly on monocytes, right? And actually, that's the only change to workflow there. So hopefully addressing your question there. And for what it's worth, that blocking solution blocks both the binding of any of our labels, this background binding of any of our labels. It also blocks the binding of any of the PE tandems in that background there.
Does that blocking solution also block FC receptors, or do you need a separate blocking for that?
It's a great question. Yeah, you need a separate FC receptor block. So this is purely for, if you will, the stickiness of some of these fluorophores. Yeah, it doesn't block up the FC receptor. So if you're in mouse, you're going to come in with a 2,4-G2, or you're still going to have to do an FC block step. Yep. So I think we've got two different questions about DNase degradation. And so the answer there is on DNase is that the DNase, actually it's interesting because it actually doesn't affect the phyton, the NovaFluor structure itself, that cruciform. It will, however, cleave, in high concentrations, it'll cleave the NovaFluor antibody conjugate. So we do have a number of people who have used these in tissue preps. So what we just recommend is that you wash out, for instance, if you're doing like a DNase collagenase tissue digestion, that you just wash that out significantly, because at low concentrations we don't see an impact to staining after that DNase has been washed out. And so the next question I've got here is, so to clarify, can these fluors be sandwiched in between other fluors, or are they replacing? How would one do this on setting up a Fortessa with Diva software? Yeah, it's a really good question. So I think there's two different answers there. One is, particularly when you're thinking off of a Fortessa, the way we've worked with folks off of that instrument or something like a Fortessa or a Symphony is particularly if you're already at the limit of what you can do on terms of the detector array, what we would say is, look, the conversation really should be, hey, what can we remove in terms of some of these APC and PE tandems so that we can go in and hit every single detector uniquely as if it were a pixel, right? And so we've done this. I think my favorite example there is what we did on a FACS Symphony, where we went in and replaced PE Dazzle, and we were immediately able to put an additional 3 colors into that, given that detector array, right? And it's this and that's on the yellow and blue. It's exactly the same story on the red. So I'd encourage you there on, be happy to engage and say, here's the fluors that I'm using, and this is what could be possible off of your Fortessa. Would love to get into the details there. Have you considered using locked nucleic acid for making your dyes? That's a super interesting question. No, we haven't. And so I think it's interesting. I think a lot of there's a lot of different types of nucleic acids we could use here. And in fact, there's even nucleic acid that are sort of have a fluorescence characteristic on their own. We've been really focused in on leveraging it as a physical platform where you can engineer this fret, and have been working along that line of logic. It's a super interesting question. Haven't considered what you're saying. Good question. Next question is, can you use them as fish probes? Yeah. Yeah. So the answer is yes, because one of the interesting things about these is that you can essentially swap in the sequence that they have off of the end of the Phyton, and you could leverage those for genomic targeting. We haven't done any specific work on that yet, but we know that they'll hybridize in solution, right? And it's actually very easy to swap those in and out. So I'd say we don't offer those commercially, but it's something that's very much of interest to us. So I'd say there, we'd love to talk about a collaboration on how you could leverage these for that kind of targeting. Yeah, and then similarly, I think maybe related to that too is some of the work we were doing in imaging. And so, you know, some of the labels that we've been testing in imaging, particularly off Nova Yellow, are very, very bright in imaging. We've been optimizing, honestly, in line with some of the intracellular experiments. We've been optimizing the background staining to make sure that you're getting, you know, better staining there. So we're working with a partner on that because we're clearly a flow shop, not an imaging shop. And so we're hopeful to have an update on that before the calendar year end, and also some recommendations for like, okay, which labels would you use in FFPE, which would you use in frozen, what would the protocol look like? So hopefully driving towards that by the year end. Yeah, good question. Are you adding these to FluoroFinder? Yes, they should be. We've been working with the FluoroFinder guys. They should be up on the general FluoroFinder spectral viewer. And we've been working to make sure that all those new labels are also up so that we can look at the spectrum of those dyes. I actually don't know the status of that because we were quite literally talking to them at the end of last week to make sure they were in. So now I just got to make sure I follow up and that they all live up there. The first 6 labels I showed you are also up on the Cytec Aurora spectral viewer. And then we do have some data, and we're building out essentially 4 profile pages on our own website as well. That'll have that data.
Any other questions for Mike?
There don't seem to be any more questions, so I think we are ready to wrap up. I want to thank Mike once more for the presentation, and for more information you can see his email on the slide, mike.stadnisky@phitonex.com or catherine.friend@phitonex.com, and general email info@phitonex.com. Thank you so much for attending the presentation, and this webinar will be uploaded on the FlowTex YouTube channel within the next few days or max 1 week, I would say.